Good morning, all. As yesterday, few of you has requested for explaining about the safety measures in laboratory. Just I would like to discuss those few things. Then I will discuss the general problems what we handle in immunology. So we don't have funding and we don't have kits and uh, at MSc level, the student should be thorough with the without kits also. So that I will deal in the second there's after completion of this one. And some queries, some questions have been asked by mail and WhatsApp to me. So I will answer after these safety measures. And let me actually, this is a video presentation because I would like to show you the good infrastructure which I collected from the Western. Actually, few videos which are having the audio in it are collected from some other sources, and the videos without the audio are of our creation. So just we'll see this one. What are the lab safety measures that we should take in microbiology? Because these microbiology teachers requested this aspect. But when almost these measures, if you can take, you can handle all other laboratories also. So we'll see the so
Dealing with a laboratory emergency. Primary containment. Primary containment concerns the protection of personnel and the laboratory environment from exposure to infectious microbes. Proper microbiological techniques, such as the safe transport and disposal of cultures, along with the correct use of personal safety equipment, such as gloves and safety goggles, go a long way toward accomplishing the goal of primary containment. Secondary containment. Secondary containment deals with protecting the outside environment from exposure to infectious organisms. It depends principally on the design of the laboratory and the availability of equipment. As well, laboratory workers should maintain the lab's safety features, such as keeping a closed door closed or leaving an exhaust fan on. Prior to the lab. Dress appropriately for the lab. No open-toed shoes or sandals. And avoid clothing with baggy sleeves that could catch fire or hinder your movement. Know the location of the eye wash, safety shower, fire extinguisher, and first aid kit. Take a moment to learn their operation. During the lab. Always wear a lab coat, gloves, and safety goggles while working in the lab. The lab coat should only be used during lab and should remain in the lab. Even discounting potential biohazards, a lab coat will protect your clothing. There is a reason many of the chemicals you will be working with are called stains. Wash your hands prior to beginning the lab and just before leaving as well. Tie back any long hair. It is both a source of contamination and a fire hazard. Disinfect your bench top with a disinfectant prior to beginning work and just before leaving the laboratory. Disposal of contaminated materials. Dispose of plastic petri dishes, swabs, disposable gloves, inoculating tools, and similar non-reusable items in the biohazard container. Reusable supplies, such as culture test tubes and glass pipettes, should have all labels removed before being placed in a rack or container designated for autoclaving. Used microscope slides should be placed in a container for autoclaving or soaked in a disinfectant solution for a minimum of 30 minutes before being discarded. The international biohazard symbol on the containers not only marks the contents for autoclaving prior to disposal, but also cautions anyone in the room as to the possibly hazardous nature of the items inside the containers. And remember, do not overfill the container and never force objects into the container. Safety considerations. If you are pregnant or if you feel you shouldn't be in the lab because of health concerns, candidly discuss with your instructor. If your skin is exposed to microorganisms as a result of a spill, immediately wash with antiseptic. In the event of a spill, notify your instructor and your lab partners immediately. Broken glass and bacterial cultures are a hazardous combination. With your instructor's approval, cover the spill with paper towels and saturate the towels with disinfectant. After 15 to 20 minutes, carefully wipe up the spill and discard the paper towels in the biohazard container for autoclaving. Discard the broken glass in the sharps container.
Hello, my name is Liz Helson.
And I'm Sherry Roberts, and we're here to talk with you today about safety in the laboratory.
The laboratory experience is an important part of this course, and the following recommendations are intended to help you develop good laboratory work habits and keep the possibility of accidents to a minimum.
So listen carefully and ask your instructor any questions you may have.
First, we are going to talk about the exits and safety equipment present in all the labs. Your instructor will show you where each of these items is located in the lab you are using. There is also a map showing these features on the bulletin board in your classroom. Be sure to familiarize yourself with the locations in the lab. These are the exits to the main hallway. If you are asked to evacuate the lab, these are the exits you should use. Here is the fire extinguisher. Each fire extinguisher is a bit different, but the instructions are clearly marked and involve removing a pin, aiming the fire extinguisher at the base of the fire, and squeezing the lever to release the foam. If you find your clothing on fire, do not run to the fire blanket. Have someone else open the cabinet, remove the blanket, unfold the blanket, and wrap it around you. You should then roll on the floor to extinguish the flames. Have someone call public safety using the emergency button on the lab phone. If you get something in your eyes, remove your contact lenses. Move quickly to the eye wash station. Put your eyes close to the faucets. Turn the water on. The on paddle is to the right. Hold your eyes open and roll them while rinsing them. Rinse for 15 minutes. Ask someone to call public safety using the emergency button on the lab phone. Here is the first aid kit. It contains standard first aid materials. Please notify your instructor if you need to use the first aid kit. Children are never allowed in the labs and cannot wait unsupervised in the hallways. We know this can be hard, but please make childcare arrangements for your children before coming to lab. Don't work alone in the laboratory. Your instructor can give you the schedule for lab monitors. Pay careful attention to any additional safety precautions given to you by your instructor at the beginning of lab. We're going to talk with you today about additional safety procedures in the microbiology laboratory.
The laboratory experience is exciting and interesting. However, you do need to know that improper handling of equipment or microbial cultures can result in injury or infection. There is no smoking, eating, or drinking in the lab. This includes gum and throat lozenges. You must never bring any food into the lab, even if no lab work is being done. You may, at your own risk, leave food and drink on the table outside the lab. You must supply your own lab coat and wear it for all laboratory exercises. Write your name on the front of your lab coat and keep it in your assigned drawer. Your lab coat must be autoclaved before you take it home, or if it is contaminated. You must wear eye protection when you are heating any chemicals. Before using the goggles, clean them with a safety clean towelette. One example is during steam staining. You must also wear gloves when you are steam staining. Tie back long hair during any laboratory exercises. You need to dispose of contaminated material properly. Pay close attention, as this is extremely important. Put all culture tubes on the racks on the lab cart in front of the room. Be sure the caps are loosened. Tell the instructor if the racks are full. There are plenty of racks. Do not leave tubes in cups on the cart. Do not put tubes or slides in the autoclave bags. These are for plates and gloves. Put all plates in the labeled autoclave bag at the front of the room. Put all contaminated paper towels and gloves in the autoclave bag in the large labeled red bin in the front of the room. Put all heat-fixed stained slides, broken glass, or other contaminated sharp materials in the broken glass pan on the lab cart in the front of the room. Do not put them in the autoclave bags for plates or paper towels and gloves. They might puncture the bag. Miscellaneous things like contaminated cups and pasture pipettes that will be reused should be placed in the plastic bin on the lab cart in the front of the room. If you are unsure of how to dispose of contaminated material properly, please ask your instructor. Before and after each lab period, pour a small amount of disinfectant from the brown bottle on the lab bench onto the lab bench and wipe down with a paper towel. If you run out of disinfectant, the supply carboy is on the sink at the back of the room. Sometimes spills occur even when you're trying to be careful. Here's what to do. Cover any microbial spills with a paper towel. Soak the paper towel with disinfectant from the bottle on your bench. Let the soaked towel stand for 20 minutes. Use gloves to pick up the towel. Place gloves and towel in the large red biohazard bag in the front of the room. Transport tubes and store tubes in incubators, refrigerator, or lab bench in cups or racks. The cups are in a cardboard box on top of the refrigerator. Keep plates in the incubator or refrigerator, an area designated for your class. Do not store microbial plates or cultures in the drawers. Learn how to light your Bunsen burner correctly with the striker. If you have trouble, you've tried more than 2 times and the Bunsen burner won't light, turn off the gas, check the striker to make sure it is making a spark, check to be sure the valve at the base of the burner is open, and try again. Always wear gloves when steam staining. Other times it is up to the instructor. Let your instructor know if you have an open wound on your hand. Dispose of all waste from staining procedures properly. Waste containers for acid-fast staining are in the sink at the back of the room. Wash your hands thoroughly with soap and water after handling microbial cultures. Also wash your hands before leaving the laboratory. In fact, you should always wash your hands frequently, and especially during flu season. Rub your hands together under running water for about 20 seconds. You can sing or hum Twinkle Twinkle Little Star. That takes about 20 seconds. If you are pregnant, immunocompromised, or taking drugs that suppress your immune system, please talk with your instructor. Lab time goes by very quickly. Please come to class prepared. Read and underline key points in the introduction and procedures part of the lab. Complete any pre-lab assignments. If you come to lab unprepared, you are more likely to be confused, make mistakes, and not complete the laboratory exercise. Now that we've gone through all of the safety procedures, please complete the information on the safety sheet your instructor has given you and turn in the bottom part to your instructor. You are now ready to have a safe and I hope educational and enjoyable time in the laboratory section of your course.